Okay, let's go ahead and get started. Today we have with us Wells Yu, and he's going to be talking about network medicine in the age of artificial intelligence. So join me in giving him
Yeah, thank you for inviting me to just present my research work in this AI at AU community. Little introduction about myself. My name is Wells Yue. I'm an assistant research professor at the Health Outcome and Research Policy Department in the Harrison College of Pharmacy. My research focuses on the complex disease research, including biomarker discovery, functional genomic analysis, and personalized medicine by developing systems biologies, algorithm, and machine learning models. So the today's presentation, the overview is going to go through the background, approach, conclusion, and discussion, and future direction. So perhaps you may wonder why we are transitioning to AI-enabled network medicine. AI-enabled network medicine offers a personalized, highly sensitive, and robust approach to both new drug discovery and drug repositioning. It reviews molecular mechanism in genotype to phenotype association. Ultimately, it aims to revolutionize the drug discovery by the goal of identifying the right drug for the right patient at the right dosage and at the right time, the 4R here. There are two major challenges remaining to overcome. One is the misclassified treatment plans with AI informatics that we need to conquer, and the other is the dynamic response. They always just get the acquired drug resistant, so we need to refine the plans based on the response of the molecular profile from the patients. In terms of the big data source, we have a big data analytics to pave the way for personalized medicine, which include patient omics profiling. Like the patient, we want to see all the available data from the big data sources. So there's three major categories of the data source. And we also want to use this data to do the data stratification, improve the biomarker with the diagnosis. So it's always to answer the yes or no question, and the prognosis is to answer whether the patient will become moderate or severe as the outcome prediction. Traditionally, we have the integrative gene site network and pathway analysis known as GNPA. We are going to characterize the large omic data sets in their complex biomolecular system context and perform this GNPA to improve biomedical discovery potential. So here is the workflow of this. Usually the upstream analysis will have those traditional data extraction, noise reduction, and statistic analysis, in which we're going to see this process is automated and very mature using the CPU intensive calculation. You can just do that. And in the downstream analysis, we see the web app validation and do the publication and share. But in the middle part is the integrative omics analysis is relative time consuming, also many fragment tools to be seen, and also specialized biomaticians always need to be required in this process. And the tools like we always see the clustering, network analysis, pathway analysis, and gene set enrichment analysis is relative like fragment and immature. So the next generation of GNPA, we're going to review the novel signal pattern and in the complex disease, especially to do the new disease biology hypothesis to find the robust biomarkers to form the new therapeutic targets and do the new precision medicine recommendation. Where we have seen this workflow, the first part is the patient stratification. We use the GETC candidates, which get the candidates from the genetic mutations, differential express genes, drug targets, and clinical data in EHR. And the second major part is molecular mechanism identification, which we have developed tools such as Wiener, Viper, Beer, and DEMA. We're going to review the molecular mechanisms in the network view and also find the most biologically relevant pathways here. So the integrated AI for network medicine are going to see this process. Like the first step is to identify the cohort. We have developed the seeds. And to explore the molecular signal pattern, we have developed the Pager, Viper, and Winner. To screen the candidate drugs, we have developed the Beer, and we also have offered advanced options like to visualize the signal pattern using gene terrain, or to do the signal pattern discovery using the secretion signal map, or we can, if we have the Medicare claim data, we can perform the health-related quality of life regression-based ensemble learning model. And for the single cell that become more popular, we have the single cell functional genomic analysis. So in the first part, I'm going to introduce the traditional one with the three major steps. So the two here is a statistical enrichment analysis of samples, also called SEAS, is going to find which clinical types or clinical attributes are enriched within a sample subset, which we have this conceptual framework. We can do the clinical type exploration and also cohort selection by seeing the foreground and background. So foreground here is marked in green dot, and the background is marked in the orange dot. And by using this selection of the foreground, we can pull out the enriched discrete clinical types such as age, chemotherapy days, and as well, we can also do the continuous clinical type, including the days to death and so on. So by using these SEAS software, we can specify the clinical type for any selected cohort and find the patient similarity based on their embedding of the clinical types, and also do the patient cluster and see whether we can find very meaningful cohorts that with the clinical type underneath. So in this scenario, we have found that if we use the chemotherapy resistant as a foreground, we can see a severe drop of their survival curve comparing to the background of the orange line. In the second tool, we're going to present the pathway annotated list and gene signature electronic repository, which we call the Pager. It's the major goal for this is to do the functional genomic analysis. We have built a server for this. So first, we have a standardized naming convention. We call it PAG. The PAG consists of the 3 letters plus the 6 digits. The 3 letters stand for the type of data, derivation method, and relation details. So by directly see the letter combination, we can identify what the source, what the kind of the type of the data can be explored from our database. And the current data coverage in Pager 2.0 we published 2 years ago. We have collected all the human genes and gene-to-gene pairs, and significantly, we have discovered around 84,000 pegs and also over 7 million peg-to-peg pairs. And mostly it's come from the M type, which I will introduce in detail about the co-membership type of the peg-peg relationship. So for the data quality control, we're going to introduce a metric that can identify what the cohesion, like using the gene-gene interaction to measure the cohesion of the pegs, which we have a co-I to indicate which the foreground interaction is overrepresented than the background. So by using the 4 number, the count of the foreground, like this circle is representing the peg, we're going to count the interaction in this peg, and another number is the theoretical interaction among these count of the pegs, and the background will be all the count of the interaction, like marking this light blue, and also the theoretical interactions we can use. And by using this 4 number, we can come up with the co-I score, which we use negative log of the p-value from the hypergeometric test. Similarly, we also count the triangle here. So the foreground triangle, we were going to see the dark blue here. The foreground is 3. And theoretically, we can also form how many triangles in theory to be in the foreground. And same-wise, we count the background triangles and the theoretical counts of the triangles to come up with the p-value also in the hypergeometric test, and therefore we can form the co-I and co-T, and we sum up together to get the COCO score. It's called the cohesion coefficient. And with the larger of the number here, that means in the peg, within the peg, we have observed more triangles or interactions than the background. That means it always have a biological relevance using the network view. So for the peg-peg relationship, we have introduced 2 types of the peg. One is called co-membership gene set network, which very straightforward. We're going to count the overlap between the 2 pegs, which we counted as small k, the shared genes. And we also have the capital K, the number of the sites in the gene set B, and the small n means the number of the genes in the gene set A, and the capital N is the total number of the genes among the A and B. So altogether, we have used these 4 numbers together to perform the hypergeometric test to give a score, like how strongly these 2 pegs have connected with the shared gene members. And for the regulatory gene set network, we have inherited information from the gene-to-gene regulation, which we have used a triangle to indicate that. So by seeing the upstream PEG A and downstream PEG B, we will observe more of the gene regulation from A to B than the background, like the gene from A regulated outside A, and the gene from outside the B regulate the genes inside the B. So therefore, we can also form a kind of the 4 number, the to calculate the hypergeometric, to calculate p-value from the hypergeometric test to evaluate the regulatory relationship between the 2 pegs. So therefore, by using the pager, we can have this standardize the peg and the quality control to say whether this peg have a biological relevance, and also form a network using the peg-peg relationship. The third tool is called winner is weighted in network node expansion and ranking. So here is the framework and with an example here. So the major function here is to do the node prioritization based on the topology. And we also count the weighted edge to be contributed in the node. So we will see that a node in the hub always have a strong correlation to other neighborhood. And also if this the strength of the connection is bigger, the prioritization of that node will be higher. And also we have the option to do the expansion, where we will also see this like the hollow nodes in the background. And once we have the nodes that have a strong correlation to the city network with a very good connectivities, therefore we can give a score here. So in this principle, we can also evaluate how strong this extended node connect to the seed network. We also carry out two significance tests. One is to compare the extended node based on their local connection, and the other is evaluate based on the comparison to the similar seed. So if this node have a connection scenario similar to the hub node, it will be very significant using this test. So combine this together, winner can rank the topology in important genes with the statistics, and also to infer the important expanded genes with statistics. The fourth tool we call the Viper is for the weighted impact edge ranking. So we have established a good prioritization of the genes. So we have shifted our idea to convert a network in the edge to a node. Therefore, we can also apply the same principle to prioritize the edge instead. So firstly, we do the Distra-like algorithm to get the optimal connection from every node, and to build this, we call it the distance-based graph. And use the edge-to-edge distance, we can expand every node here just stand for the genes, but in this one, every node stands for the edges. And to expand this to an edge-to-edge distance matrix, we call it a traverse path network. And by building this network, we can perform the ant colony algorithm to do the prioritization and do the p-value estimation. And therefore, we can prioritize the original edge in the graph to see which is very important in this. So how does this perform? We have observed that if we have used a traditional edge prioritization, we have the very high resolution to distinguish those edges, such as in this subgraph, we have two sub-network connected by the edge. So theoretically, this edge will be the most critical edge in this. So therefore, we can see that mostly all our compared algorithm will rank this as the toppest. But we have performed better to identify the sub-network connected edge, like the green one is better than the blue one that cannot be discovered in the traditional way. So therefore, Viper have a good identification globally to find the edge in the topology structure. The fifth tools we call the biomedical entity expansion, ranking, and exploration beer. Is for the semantic network of the biomedical entity. So we have this pipeline for the beer to do the data processing. So we have the Medline processing, original the data from the PubMed. We also do the natural language processing to mine the semantic relationship and we also do the graph summary of this network. So the predication extraction always consists of this subject, object, and the predicate. So where the subject always kind of the gene, object is some of the like the concept or biomedical terms we have observed, and this is a verb in the semantic relationship as a predicate. And we also do the unified medical language system to get every entity to be normalized. And the predication filter and ranking, we can just form this kind of the semantic relationship. And finally, we can build the concept of network where we can just see the different type of the predicates with different color in the network. The data source for this beer, we have the gene-gene regulation or gene-gene interaction from the HAPI database, which we have summarized all the available source and assigned the score, like summarize the final score from different sources using this formula. We also have the semantic relationship that as mentioned in the SAMMED-DB, and we also have the peptidator for the validation of the protein-protein interaction in our beer database. So currently, we have covered over like around 3,000 quality ranked protein-protein interaction, also over 19 million predications with all those gene to PubMed ID relationship around 7 million, and all the predication to PubMed citation is around 96 million. So what can this beer do? We have like a conjunction analysis in the GBM study, which we can get all the genetic risk from the OMIM database, and we populate all those genes from this as input. We can validate the gene symbol. Some of the gene symbol may not use the official gene symbol name, therefore we can convert that. We also can do and extract this protein-protein interaction either from our HAPI database or from the semantic relationship from the literature, and we can do the ranking and also extend the field, like not only use the genes, but also use a biomedical term, including some drug or other concept if you're interested, to build a network and also show the overall distribution, and also use the frequency to build a word cloud to see which one or which concept is most significant in this network. So as you can see, we have built these 5 tools or established these 5 tools to do this network biological analysis. So in the case study of the glioblastoma disease, as you may know, glioblastoma, also known as GBM, is an exceptional aggressive brain tumor that affects around 17,000 Americans annually. It's a deadly disease with a median survival of just over a year, like 14 months. It is quite difficult to treat because it's not surgically curable, since the complete cut of the tumor is extremely difficult. Also, the tumor is quite resistant to our conventional adjunctive therapies like radiation and temozolomide. It's highly invasive. With this, like in mind, this research had been carried on for a long time. Despite like the 75 years of the research, we have essentially at only 1 month survival per decade. So part of the failure is due to the severe limitation of our preclinical model system. Like we have used the advanced age, like over 75 years old, with a severe comorbidity, and also the blood-brain barrier is specialized the nerve vascular unit involved to maintain the brain homeostasis. So therefore, this is draw attention to the research challenges. There's a research to find the subtyping of the high-grade gliomas. They have identified these three major subtypes: the preneural, mesenchymal, and proliferation. So for the original the HGG samples, they have identified 108 differentially expressed genes, and they narrowed down the 35 signatures genes to find the clusters that can identify this particular subtype. And within this survival analysis, they can see that preneural comes with a long survival, and mesenchymal comes with a shorter survival. Proliferation, we always have a severe scenario with the shortest survival as seen here. Another motivation is to see the clinotype leading to the subtype specific gene signature, which there's a publication to see that if they use the t-SNE or UMAP to do the dimension reduction, they can always see that if they want to use a clinical attribute called the smoker status, they cannot find a unique pattern to distinguish. So it's universally distributed in the map, but once they change to a gender look-like, they will see a very clear pattern, like men have a blue dot here. Use t-SNE, you can see there's a clear margin to distinguish these two gender. As well, the UMAP also have the same thing. So therefore, they can perform like a k-mean clustering to find a specific cluster corresponding to the gender, and therefore they can form this gender-related or gender-specific biomarker to identify why this phenotype have a low survival than the others, and therefore they can have a precise description about this gender-related subtype. So inspired by that idea, we're going to build a multi-omic GBM data set and to find survival molecular subtype biomarkers to potentially increase the survival time from particular subtype. So we have collected data, the RNA-seq samples from the TCGA database. The multi-omics, we will have the genetic mutations, which we collect around 540 genes. We also have the differentially expressed genes and the drug targets. As well, we will involve the survival clinotype here. We particularly use the disease-specific survival known as DSS, the unity days. And unify those candidates together, we have a list of 911 genes in total. We also introduced a refined weighted embedding for clinical convergence. Specific, we called it in-cluster mean of the square distance, MSD, to measure the degree of the convergence. So the idea is very straightforward. We have used the weight, like weight vector, to multiply the embedding vector here, and we can form this refined embedding, and to see whether the foreground, the blue part, is cohesive enough than the background. And to minimize that, we will go through all kind of the weight vector to try and to minimize those MSD index. So in practice, we have the molecular subtype along the weighted survival time projections, where we have this ISMB, and we're going to see whether the survival curve, the survival and the subtype can be distinguished. Therefore, we can see that there's a survival high and survival low pattern from different subtype of the mesenchymal, preneural, and classical. And we can perform the pathway analysis using that, and giving this traditional pathway, pager can popular all those network and to find the higher level of the concept using this right color, especially for the cancer-related pathway. We have get an example of this pathway. This is a glioblastoma signal pathway. We can form the subtype-specific gene panel and found this survival curve. The significance of this research, we have found the survival signature pattern combined together always outperforms any individual genes here. As well, in the enriched clinical type confounders, we have performed the seeds to find survival-related covariance. And always we see that this covariance is always related to their survival time. Like PFI is the progression-free interval, OS is the overall survival or disease survival, specific survival or the disease-free survival. And it always have a good identification of this survival using those different biomarkers in this subtype. So with all those information we get and prioritize using the network view, we can reconstruct a signaling pathway using the pager database, happy database, and also the signals from particular subtype. So this subcellular location is using the probabilistic infer from their neighborhood. So we have this information from the gene ontology cellular components, and it always tell you where the molecular travel from the subcellular space. For example, we have the major membrane here is extracellular membrane, intracellular membrane, and nuclear membrane. And we can see the traffic of the gene signals enter this different membrane. We also have collected those mechanisms like activation, inhibition, phosphorylation, or it's just a transcriptional regulation or interaction mark in this pathway. And we can see that with the survival high mesenchymal group, we can see there's a downregulation of these two genes, which is very different in the expression direction in the survival low. As well, we also see the perneural classical to find how these genes talk to each other in the subcellular space. Another thing is, once we have discovered all these very critical biomarker, can we kind of validate using the existing publication from the well-known literature? Yeah, we have developed this beer to find the evidence of the candidate gene signatures, novel genes, and generate new hypotheses. So this is a conceptual beer for the GBM analysis. We can form the receptor signal pathway with the receptor ligand binding here. And we have the signaling pathway modules like the PAG inside of this subcellular space. We have a validate, can be direct validate with the upregulated panel here using the GBM, but we also can infer some indirect evidence that can be very novel. So the pipeline of this beer validation, we can retrieve the traversal path covering the network-based survival gene signature first, and then build a beer query list using the gene symbol, full name of the genes, and the glioblastoma as a disease name. We can retrieve all the semantic relationship and construct a bioentity semantic network by using these entities. And in terms of the novel genes, we continue to expand to infer the potential functions related to the GBM. By using that pipeline, we can go into extract the biomarkers from the past and to see whether we can kind of find the literature to support these gene markers is important in the survival of the GBM subtypes. So there's something that to be very novel that haven't been fully reviewed or the functional related to the GBM, like these three genes, EP300, FOX4, CDKN1B in this mesenchymal, the lower survival patients. We have found that some indirect evidence to see the EP300 the mutations observed in the Robertson-Darby syndrome. Also has seen that FOX4 is related to hypoxia responses, maybe infer some inflammatory inflammation mechanism underneath this mechanism. Or the CDKN1B, the loss of these can cause the neuroblastoma diseases. Moreover, with all the multiomics data in hand, we can review insights from the interlayer correlations. We particularly find a lymphocryptovirus, a potential microbiome involving in this CDKN1B gene. We have the upper layer always come with a copy number alteration, methylation, gene fusion. In the intermediate layer, we have the RNA-seq data, which we have construct this pathway information. And we can also find this microbiome data with the microbiome expression and kind of infer the correlation between the RNA-seq and the microbiome. We can build this logic. So if we observe that CDKN1B is high, is always block the lymphocryptovirus, and therefore this can potentially lead to a low survival in mesenchymal. Right. So once we have this, we always want to inhibit this. So if we observe this CDKN1B, our potential treatment is going to bring this high to block this lymphocryptovirus expression to potentially increase the survival rate of these mesenchymal patients in GBM. So the conclusion and discussion for this part is we always come with the specific 3-step. In the patient stratification, we always provide compiling evidence for putative mechanism or gene modules underneath the poor survival subcohort. And also we have use an example of the cancer-related pathway for the glioblastoma analysis. Also observed like PI3K trigger events or FOXO family signaling pathways in mesenchymal and perneural subcohorts is pretty important. And for the survival signatures, we have reported panels specific to each subtype, and also observe these panels always outperform the individual gene components for the survival prediction, and very promising for future clinical application evaluation. In the multi-omic data integration, we can find a lymphocryptovirus is important as a microbiome involving the disturbance of the CDKN1B gene and lead to the low survival in the mesenchymal GBM patient. And next part, we're going to see the advanced options with the visualization technique called gene terrain, or the secretion signal map. Or if you have the Medicaid claim data, we can perform the drug screening to see the potential impact of the drug pairs related to the health-related quality of life, or we can do the single-cell functional genomic analysis. So first of all is to see the gene terrains have to visualize the signals that group together. So the input usually come with the gene expression profile, gene-gene interaction profile, and a gene coordinate profile to generate a map. With this map, we're going to use gene terrain to get the flooding modules implied from the control line. So we have a scale like here we have indicate the human customized effect scale. If we increase this scale, we kind of see this synergetic effect of the group of the genes better than the isolated genes. So you can see this group of the genes signals individually, they didn't have a good signal, but once they get together, like using this scaling factor, they kind of increase the signals more than the isolated genes. And therefore, we can find potentially a group of the genes in the panel to see they overcome the robustness problem and always give a highly sensitive outcome. So the idea is to for the layer generation using this three type of data as input, and we draw this gene terrain, and we also have a signal vectors underneath this gene terrain with a value, the pixel value, and we can use this pixel value as the embedding to generate the dimension to reduce the dimension to plot in the t-SNE or UMAP or dense map. This transformation of the GT expression matrix to a lower dimension space can help to distinguish different clusters in this map. So similarly, we have used the IMSD introduced before to see once we have used a different setup of the expression as the control, and we have used the gene terrain signal vector, the second line as the case, can we just further reduce as MSD? And we have seen that this map shows the best performance with a balanced reduced cluster S and B. As you see, this blue color is from the blue color, orange color, and green color is from the three subtypes of glioblastoma. And for the glioblastoma, the YLab always have a in vivo culture of the human GBM cells, which they call the glioblastoma patient-derived xenograft GBM PDX. The principle here is to get the tumor from human first and do the culture to get the GBM sphere. And after they validate this sphere and do the transduction and transfection to the mouse model, like and to culture in vivo to see these different in vivo validation using the histology, fluorescence, and bioluminescence. So by getting this data from the web lab, we have the 8 pairs of the radiation resistant versus the parent lines from human and the selected lines from the mouse. And if we have the treatment like a radiation treatment or temozolomide treatment, can we see there's a significant maintain of the survival time? If this is maintained, like the patient doubling time is similar to the selected doubling time, that means it's sensitive to the radiation. And for the temozolomide, we also assigned the temozolomide score, which use the control median survival versus this treatment median survival. Use this treatment divided by the control, we can get a score. If this score is larger, that means it always comes with very sensitive with the temozolomide treatment. So therefore, with those score assigned, we can see different samples with the treatment, like the 1153X is the double 1516 is the double resistant in this scenario. So by using this gene terrain, can we visualize those gene panels that correlated to the phenotype changes? So for example, we have used the color scale to indicate whether it's a radiation resistant or temozolomide resistant. So for the radiation resistant only, we have seen a very consistent of the signal pattern here, right? So this radiation resistant, but come with a temozolomide sensitive related, we have observed this part of signals have consist upregulated. But for like a double resistant here, always see a very significant drop of the expression in this corresponding pattern, and you cannot find this pattern in any other setup of the samples. So therefore, by using this gene terrain signaling pathway or signaling map, we can see a correlated signal patterns can be correlated to their phenotype as the treatment responses. And another thing is to see whether the spatial information integrated, the spatial information can be quantitatively measure the signals as well. So we have used an example from our collaborator in the material science lab. They have the cell secretion, like this is a cytokine interleukin 6, and can we identify the T cell subtype of the specific type of the secretion pattern here. So they always have like direction if we observe in an anisotropy, and if they have a universally without any direction spreads of the signals, it's called anisotropy. So for this one, we have seen that by giving the derivative of inequality index we have introduced using this formula, we can see there's a significant direction that can be captured using the DII number here. So therefore, we can see from the 10 minutes, 20 minutes, 30 minutes of like in the longitudinal analysis, we can see this consistent to be increased in this anisotropy. But once we have used the anisotropy, we see this is universally distributed of the signal without any clear direction, and therefore this number should be low. So therefore, by using this secretion signal map, we can identify different types of secretion, and this can help to be sensitive enough to quantifying the cumulative signals and the signal dynamics in the longitudinal analysis. And we have established a app here, like using this URL. So for the conclusion and discussion for the visualization using the gene terrain analysis, we can see that gene terrain can boost the performance of the patient stratification. Also, we can interpret omics result with advanced gene terrain signal. And for the measurement of the spatial information secreted signal, we can also use a DRI as an enhancement. And for the claim data, we kind of formed the idea of the health-related quality of life drug screening model in the prostate cancer. Since the prostate cancer relative not that aggressive, so the major concern is how we can maintain the quality of life of the patient. So we have observed the polypharmacy in the patient. Like we have marked in this graph to see the no polypharmacy is blue color, the moderate or the polypharmacy we just marked as orange, and the red is extreme polypharmacy, which they have take the concurrent drugs over than 10. And therefore, we can see the distribution of that is equally, right? So we have seen a lot of polypharmacy in real world case. And the next thing is to detect the deleterious drug-to-drug pairs, and find whether it has a high impact of the on the health-related quality of life. And we have observed the trend that without polypharmacy, they always maintain a good score, but with extreme polypharmacy, which had the lowest score. And by build a embedding space, we can see the physical function have be the low score of the physical function have always located in the upper left region, and therefore we can do the selection of the severe patient, which can be marked in the right area, to as a foreground to pull out those drug-to-drug pairs severely impact to this low quality of life patient. Incorporating these clinical or demographic characteristics into the health-related quality of life, we can see the mostly related to the polypharmacy or their functional lower score related to the age, BMI, comorbidity, and income. So this blue region means it have a physical low function, and this blue region in this graph means the low function in mental. And the polypharmacy, the extreme polypharmacy is the upper left region, and we see there's an age distribution. Some of the older adult here in the prostate cancer, they have severe polypharmacy and always corresponding to their low functions. But there's also some of the high or older patients without this polypharmacy that always keep a good health condition. And for the BMI as well, BMI have a more correlated to the physical function other than the mental function. And the comorbidity is also always located here in the upper left region. Therefore, we can see this highly correlated to their polypharmacy. So with more of the comorbidity, they have a tend to get more polypharmacy, and they therefore lead to the physical or mental disorder. And for the income, we can also kind of see that the lower income cohorts always located in the upper left region. Let's select the severe cohort. Both mental and physical function, we can see here is always located in the upper left region, and we can form the drug-drug interaction network based on the hypergeometry test to see whether we can find some important drug indication in this for quality of life patient cohorts. And for the drug screening for these severe impaired patients, we have observed that the pain relief highly related to the physical function, anxiety related to the mental function, and respiratory disorder related to the physical function. All treatment combined together will imply a lower physical and mental functions. So we have this gene pair, it's a drug pair with drug A and drug B. And we also popular those background, foreground counts and calculate the odds ratio. And especially we have used 95% confidence to get the lower bound, upper bound of the odds ratio. And if we get the top ranked with always the lower 95% of the odds ratio rather than 1.4, we can get these 6 drug pairs. And we also kind of assign the number here to indicate what this drug major treatment is major treat for what kind of disease. And if we observe this as a different treatment, it always comes with both physical and mental impaired. And as well, if this is like a sole one for any one of the category, like this hydrocodone bite with acetaminophen is treated for the pain relief, and it only related to the physical disorder, and therefore we can see this having tremendously decreased than using any one of the drug. So by using the pair of the drug can have a synergetic effect to further reduce the quality of life in this particular patient cohorts. So therefore we can plot this graph, the first without the shade, that means it's a physical function drop. The control is the gray color, and the case is taken. These two drugs concurrently will have a drop significantly. And therefore, we can see all the pairs have the same scenario of this tremendous drop. But with any single of the drug, even though we can give a significance test to evaluate how significant of the drop, but the measurement to see the decrease is not that quantitative enough to as compared to the pairs. As well, using the idea of this drug-drug combination, can we capture the association of the additional drugs in these pairs, especially for the intricacy that cannot be pulled out by using the traditional odds ratio? Can we see the weak impact drug combination always boosts the performance? We have used a 19 algorithm in the 7 major categories, including the simple one like a linear or generalized linear classifier, up to the very complex like a neural network-based classifier. We found that the GBM is a gradient. Is a great the GBM is a sorry. Oh, sorry. Yeah. So is the light GBM is a LGBM is a light gradient boosting machines performed the best. And if we use the best model to do the regression, using this PSC and MSC, using the norm baseline with all the clinical features we have carried out in the previous the pharmacy, age, BMI, comorbidity, and income. Together, we can do a prediction with the R-square. And if we have added 9 drugs we have pulled out from this severe patient cohorts, we can slightly increase the performance, but with the foreground with all the polypharmacy cohort drug-drug pairs, we can pull out more drugs, like 88 drugs, and combined with the baseline of the clinical features, we can even more get a good performance by using the GBM model. In both physical component summary score prediction and mental component summary score prediction. And we also evaluate using all we know as the measurement, like from the mean absolute error to the root mean squared logarithmic error. So the conclusion and discussion from this part is we have used this HRQL RAM for the first time to unreveal the intricate association between the drug pair and the class pairs, and related to the quality of life, and considering the different demographic background. As well, we have this to potentially predict the forthcoming data set to paving way to predictive analytics in the polypharmacy. And as well, this platform can give a real-time analysis, enabling the tailored intervention in prescription and description to enhance the patient outcome. As well, if you're interested in the single-cell analysis, we also developed the single-cell functional genomic analysis, which we have used a SciSeq data and to do this platform. So this is major discovery from the mouse nature killer cell to find the differentiation or maturation of these cells come with the markers underneath with the pattern. See the pattern expression changes along different trajectory. So using this one, we can give more insights in the mouse nature killer cell function impairment and potentially give hints of what to focus in the next treatment. So future direction for the discovery using the AI-driven drug discovery, we can also go for the direction of the protein ligand docking, toxicity prediction, chemical synthetic pathway, and we can also focus on the interpretable table AI with the network, integrate large-scale multi-omic or multi-modality data, and choose the proper knowledge as a presentation in gene terrain, and solve the drug discovery problem by targeting the druggable molecular network or signal patterns. So this is the tools and algorithm we have developed so far. And if you're interested, you can just take a screenshot and you have a free access to any of these links here. And we also have the AI Farm Research Group here with the GitHub project repository. So I want to thank all the contribution from the different departments from AU and also external institute from University of Alabama at Birmingham and Indiana University. Thank you. I'm glad to take any questions. Okay.
I'm talking about many interactions between hydrocodone.
Yes. Yes. Like fentanyl and hydrocodone, these are like pain relief the drugs. And if we see there's a concurrent, I don't know why they have a concurrent, but it seems that pharmacists have prescribed these two drugs for the same patient, and it have a severe impact of their quality of life, especially related to their physical function. The physical function will tremendously drop than taking any one of the drug in comparison. Yeah, as the patient fill up the survey of the quality of life, we observe there are a very significant drop of the quality. I don't know whether they have taken action for bring up their quality of life using any remedies as well in the patient profile. But as we observe that this cohort, like by taking these two drugs together, will.
Now, the thing is, tell me a little bit more about for relations. So what does that indicate?
Oh, yes. So the different color indicates the covariant subtypes. Like if we have used the first graph here, this means the blue color means is always have a lower physical score, and the orange color means high score. So this is a trend of from high to low. The same wise, mental also have high to low. And if you see this polypharmacy, the red means they have extreme, like take 10 over than 10 drugs at a time as a concurrent drugs prescription. And this age as well is from low to high, low to high. And yeah, you mean very right, right. There's no significant correlation because we also see there's some like lower income cohorts also can maintain a higher like health. That depends more on the polypharmacy because the polypharmacy or comorbidity will have a significant impact than this income. But we also see there's some of the pattern like here, like you see a trend of a mixture of lower income patients in that region.
Thank you. Very fascinating work.
Thank you.